Um, cool. So welcome to the final lecture for machine learning for biochemical applications. My name is Clayton Kosonocky. I taught the first lecture. I'm a second-year PhD student in Andy Ellington and Edward Marcotte's lab. I'm primarily doing research in using AI to kind of annotate chemical functionality and then also predict it as well. Today, we will be talking about protein design and a little bit of protein engineering thrown in as well. So proteins perform functions. I listed off a bunch of kind of main functional groups. This includes catalysis, structural support, movement, and so on. And on the right, I'm outlining a specific example for the protein lactate dehydrogenase, which is an enzyme that converts pyruvate to L-lactate, a single stereoisomer of the product. And so lactate dehydrogenase works through the very specific structural coordination of three residues that help stabilize the intermediate for this reaction, or the transition state for this reaction. And so it does this in a very specific way where if the residues were kind of in at very slightly different positions, then it wouldn't work at all. So the whole protein, this whole giant glob of amino acids and residues, is really performing the task of stabilizing this conformational state for these three residues to then do the reaction that then performs the function in the cell. And so you can imagine also that if a mutation were to occur somewhere else in the protein that drastically changes the whole like fold of it, then it would lose the function, and then the biological cell would no longer be able to replicate potentially. So today we're talking about protein design, and really people may ask, why do we even want to design new proteins? The ones that exist work pretty well. Mainly it's because of kind of three main categories that I'm throwing out here. I have function, and so this is essentially creating new enzymes or ligand binders not found in nature. Structure, this is like creating symmetric assemblies or modifying or removing antigenic structures of existing proteins. And then I'm lumping a whole bunch of other traits into what's called property optimization. And so this is sort of more tackled by the field of protein engineering, but it is very related to protein design. And so this would include redesigning natural enzymes to work at a higher temperature. And so for actually why we would want to create new enzymes, in the modern world, we have a lot of synthetic chemicals being created, and a lot of these chemicals are sort of accumulating in our environment, and it would be beneficial to remove these and turn them back into usable bioproducts potentially. So yeah, we can design enzymes potentially to do that. And so yeah, I've kind of outlined a little bit of why we want to design new proteins, and the question now is how. And so there are multiple different approaches to doing this, but today I'll be mainly focusing on a structure-based workflow for machine learning-based protein design. And so this workflow generally follows starting with a protein function that you want, or a specific coordination of a specific functional site on the protein, and then going to a backbone that can sort of fold into that specific, or give you that specific function, and then figuring out an amino acid sequence that can then give you the backbone that you desire, and then thus give you the function that you desire. So breaking this up into two different steps even further, the first part is backbone design. And so this is really the question of which protein backbone will give you the desired function. And so by protein backbone, I'm talking about this central continuous chain that goes through the whole protein. And I'm not talking about the specific side chain. So we're ignoring that in the scope of this problem, outside of the specific residues that we want to hold constant. And the second part is going from a protein backbone to an amino acid sequence. And so this is essentially saying which amino acid sequence will fold into the structure that we desire. And so this is called inverse folding. And it's really just the opposite of what AlphaFold is trying to do. So for AlphaFold, which we talked about last class, we're going from an amino acid sequence to figure out what the structure is, more or less. And here we're doing sort of the inverse problem, which is we have a structure that we want. How do we get an amino acid sequence that folds into that specific structure? And so together, these represent a possible path to design new proteins, because we can go from the protein function to the backbone to the sequence that will then accomplish everything that we want. And so now diving a bit deeper, so today's lecture is primarily going to be a survey on various kind of state-of-the-art models that can accomplish various parts of this protein design and engineering pipeline. And so up here, I have a continuum of protein design that spans from models that excel in point mutation specifically to models that excel primarily or are capable of doing full-on de novo design. And so this is essentially starting off with none or very little of the original residues from a protein that you're trying to design, and then just generating around it to create like a whole new protein. And then in the middle, I have what I'm calling span redesign, which is basically we want to keep some parts of the protein, but we also want to modify other parts of the protein. And so one thing to note is the models that I'm going over today, most of them have been created in the past year and a half. And so this field is moving very, very fast. And a lot of the state of the art is exceptionally recent in all of this. And so the field is kind of, well, it'll be curious to see where this field will develop in another year and a half and see where we are. So first up, I'll be talking about point mutation models and this sort of approach to this on the left side of this continuum. So I think it's easiest to think about how kind of protein design works by starting off at a very local residue change, which is for point mutations. And I think as well, it's easy to think about this in terms of the amino acid sequence in a language model to accomplish this, especially considering that we talked about these language models in previous lectures. So on the right, I have just the basic schematic for ESM2, which is Meta's like big evolutionary scale protein language model. And this model was trained with masked language modeling, which is basically hiding various residues of the protein, of the amino acid sequence, and then having the model fill in what it predicts should go there. And so in this case, I have a threonine that we're selecting, and then we're masking it, and then the model predicts it should be a glycine in this one case. And so this could be viewed as a training task, but it could also be used as an inference task or a prediction task if you want. If you want to basically see like what does the model think, or which residue does the model think should be in this spot, and if it might not be an optimal residue for that original location. And so why should this do anything? Why should this model that's trained on self-supervision be able to predict a potentially better mutation? And so the real answer is that it might not be able to, or in its current form that we have up here, it's not really optimizing for any specific phenotype. It's just optimizing for essentially saying what does the model think is the most wild type, or the most like common, or it's sort of like the average mutation or the average residue that should be in that spot given the surrounding context. And so this is sort of predicting where nature messed up in putting a residue and potentially correcting it to make a more evolutionarily natural mutation, or a more natural residue. And so this, in my view, in my experience that I've kind of worked with these models a little bit, this isn't necessarily going to correlate with any specific phenotypic property, but it could actually be very beneficial in suggesting mutations that researchers might not otherwise consider. And so going back to this example, this threonine to glycine might, to a researcher, actually be a promising opportunity from their perspective to maybe optimize the activity in some way or another, or maybe increase the thermostability of that protein. And so, yeah, yes.
Just wanted to bring this up. So, just as a model generalizes, before we train, actually it should work if the model has.
Exactly.
So
yeah.
Here is this
Yeah.
I'd be curious if anybody has.
Yeah, it is interesting. I know that previously, one of the approaches was to stop the training before it reaches this memorization stage, and then trying to have a slightly underfit model that can potentially still give interesting predictions, but is not predicting the exact wild type. But it's not a very like reproducible approach for sure. I don't know. Does anyone else have any potential thoughts on this, or yeah? I meant the actual point where you cut it off. It doesn't seem very reproducible, because how do you know what the optimal point in the training is that would give you an advantageous thing versus overfitting? You maybe could like measure at various time points, but I don't know if that would generalize between different model types and architectures. or even data. Yeah. I know it was right.
Yes, and I've written. The problem here is that the test.
I'm I think it's exactly the
Yeah. I think the 3 billion model is actually underneath the order, and then their 15 billion is above the executive order. Yeah. From what I've heard. Well, it's 1,000 times less for biological sequences. Yeah. So yeah, in case anyone's curious, there's an executive order that came out 2 days ago, that basically said that if you go over a certain number of floating point operations during your training, then you have to report the model stats to the government. And we've been talking about which ones fall above and below. Yeah, it's confusing. But why did they do that? I think it's for AI safety, apparently. Yeah.
Cynical perspective, one of lobbying of the company. Allowed to do this over there.
That's like just far enough outside of the scope of this lecture where I have to bring it back to protein design. Yeah. Mhm. I think I don't think so either, unless we group together a lot of labs and funding potentially. Yeah, but
So it's certainly not something that people think. Other ones.
yeah,
But my question. How strange was that? Know what that would be.
yeah. I think that makes sense. Yeah, avoiding just overfitting for these models would be, I think, quite useful. It could also just be like the training tasks that we're doing just with mass language modeling might not be optimal, which is kind of what you're alluding to. Yes. Mhm. So that is being explored. Yeah, I think I think Danny Diaz does that at IFML. I'm pretty sure. And also, I'll be talking about in 2 slides supervised learning, so adapting these to specific phenotype tasks. And so right now we're just talking about the pitfalls to just self-supervised inference, which does have a place, I think, and could be really interesting in a paradigm on its own. Um, but yeah, so moving forward, so we previously discussed self-supervised sequence-based approaches, and now we can discuss self-supervised structure-based approaches, which follows a very similar paradigm. This is a kind of complicated schematic, but essentially the main point to know is on the far left, we can start off with a protein. We can subset this protein into a small cube or like a small sphere of just the local chemistry around a given residue. And then we can mask that residue, that amino acid, and then try to predict what was there through a model. And so this is really just masked modeling, but it's using a structure instead of a sequence. And so this is kind of a simpler version of that, where we can take that microenvironment that is surrounding that residue without the residue in it, and then we can pass it through a model to extract out the features and then perform classification to predict which amino acid should have been there. And so this approach is sort of orthogonal in some way, slightly orthogonal, in that instead of predicting based off of the sequence data, we are now predicting based off a local chemical and structural context surrounding that amino acid. It does, yeah. Right. Yeah, exactly. Yeah. So yeah, these are kind of currently their current state prone to just memorizing the existing structures that exist. Yeah. Yes. Hmm. Yeah. Yeah. I have the right. So the question is, should we just iteratively inference these models and see if we can get interesting, like kind of diverging sequences from the initial position? I don't know if there are any like I just don't know enough about that specific literature, but I do know that there is some sort of Bayesian optimization approach in, I think Francis Arnold worked on this, where you just do that without a model, and then you like have to test it every time. So you're saying don't test it every time and just diverge quickly. Yeah, right. Yes.
Yeah, think about what you want. Nice. Anyway, but what the network instead means knows the network.
Yeah. Yeah, that is true. And I talk about later on just like conformational changes for proteins and how we're basically essentially the current paradigms for protein engineering and design to treat proteins as more or less like just solid blocks that cannot be flexible at all. Um, but yeah, so moving on to essentially supervised learning. So we've been talking about predicting the wild type. The question some people may be having is, can we now predict specific properties and optimize proteins for specific properties? And so some of these include stability, activity, or expression. If we wanted to just make enzymes that can work at a higher temperature or be more active or just have more expressed in the cell. And this is sort of more or less the I think where the field of protein engineering is like slightly separate from protein design is that protein engineering seems to be more optimized towards specific property optimization or doing kind of smaller changes to existing proteins, whereas design is more focused on large scale changes. And so on the right, this is taking an approach similar to MutCompute, the self-supervised structure-based program that I showed previously, or model showed previously. And you can take that self-supervised model and then you can fine tune it on supervised data, in this case, stability data. So then instead of predicting just a wild type residue, you now predict a specific stability value or a change in the value given a mutation. So is everyone satisfied with moving on to the next part, which is span redesign? Cool. So next up is mutating multiple residues at once. And so this, I'm going back to this ESM example because it's quite simple. We can take now, instead of masking just a single residue, we can mask a whole stretch of residues and then feed them into the model and then say, what do you think should go here? And then now it has more kind of degrees of freedom and it's a less constrained problem. So it can suggest mutations that would not otherwise have been possible to suggest in a single point mutation model. So this is advantageous in some ways in that it could bring us towards a more global optima for a protein, but it could also diverge pretty rapidly and then destabilize the protein. So it's kind of a trade-off. Yes. That's a good question. I don't think this method would do that. I think the models I'm talking about are that. That's correct. Yeah. Cool. Yeah. That is a good point. You could just tokenize on a larger than one amino acid string, and then it could put in like larger stretches in a single token spot. That could be a way to do indels incorporated inside of this, which could be interesting. I believe it's just the basic single letters. Mhm. Yeah, and there's like ones for ambiguous amino acids. Yeah. But yeah, so there also are structure-based approaches toward span redesign. And this is actually an inverse folding model, but I'm sort of grouping it into this span redesign because it also accomplishes that task. And so here we can take a protein that we want, and then we can take just the backbone of that protein, and then we can figure out a sequence that would fold into that backbone. And so here I'm talking about the model ProteinMPNN, which stands for Protein Message Passing Neural Network. The name in the architecture is not quite relevant to the discussion today, but it is from the Baker Lab, who I'll be mentioning a bit later on as well. So in this case, we're not modifying the whole protein, and we're instead taking our protein of interest, and then we're going to freeze certain residues, these yellow residues and these blue ones right there, and say, do not suggest new residues for those locations. And then we can pass the frozen residues and the backbone into this model, and then the model can predict it can predict new amino acids across all of the gray spots. So wherever we are trying to predict new different amino acids than were originally there. Yes. The goal, this is trained to maintain the original backbone. Yeah, so the whole point of this model is it's just different sequences that can fold into the same backbone. Yeah. And then, yeah, so up next is de novo design. So here, I'm initially going to be talking about a backbone design model, which is called RFdiffusion. And so this stands for Rosetta Fold Diffusion. Diffusion models are these generative models. They essentially just can take random noise and then generate a structure, in our case for proteins, from that noise. And this can be unconditional, so just generate a random protein, or it can be conditioned to an initial starting point. So you could like take the active site that you're interested in, just 3 residues, and then you could generate a protein that satisfies that constraint. I'm not going to talk much more about diffusion models in this architecture because it is, I think, beyond the scope of this lecture and this course. But yeah, I think it is important to discuss this RF diffusion model because it is sort of the future of where this field seems to be going. And so I mentioned you can generate proteins conditional to an initial condition. here I have 3 examples of this. I wish I made this a little bit bigger, but here on the very top row, we have a binding target in teal that we're trying to generate a protein that can bind this specific initial target. And so here they can generate a protein, which is in pink, that then should bind that target. And in the middle, we have a functional motif that we want to integrate into our protein. And so we can take that, input it into our model, and then generate a protein that will incorporate this specific domain right there and will fold into that domain for those residues. And we can also do something if we want to do a similar thing to the functional motif initial input here. Previously, we can do that with the multimer and have 4 different proteins coordinate together to form the functional, the multi-protein motif, basically, that we want. So these are just a few examples. I'm talking about more in the next few slides with experimental validation. But before getting there, so in the RF diffusion is just a model that goes from an initial condition to a backbone. And so we can actually use the ProteinMPNN model that I talked about previously to go from the backbone to the sequence. And so this is what they do this is what the Baker Lab does in their RF diffusion paper. They probably just did it because it saves them a lot of time on having to, for one part, saves time on having to go from like the harder problem of going from noise or initial condition to the actual sequence, because they already have this model that works pretty well for this step, so they can just focus on the previous step and sort of simplify the approach a little bit. Does this workflow make sense to people, or do you want me to spend more time talking about it? Cool, I will move on. So here, I'm gonna go through a few different types of their designs in their RF diffusion paper. The first one are these symmetric assemblies that they created. And so their goal was to explicitly make symmetric protein assemblies. And so they generated 5 different ones that I'm showing here. And they actually, they first saw that these proteins, their predicted structures are in this first column. Their AlphaFold2 structures are in the second column. So they took their proteins, the sequences that they generated, and ran them through AlphaFold2, which showed to fold to a similar structure. And then they actually produced these, and then they got cryo-EM structures of them and saw that they do in fact fold into the correct structures. So these are quite different from anything really produced by nature. So they were emphasizing that point in the paper. They do, but I do not have that number. I think it was 2, yeah. I can send out an email afterwards with the success rates if people are curious. Yeah. So I do have a success rate for another application in a few slides from now, but
their method on your problem of I'm not saying that they
yeah, and I address the limitations to these models near the end of the PowerPoint as well. But so I mentioned at the start of introducing these models, or this diffusion model, RF diffusion, that we can take an enzyme that has an active site, and we can remove the active site residues in their 3D conformation, and we can then generate a protein that will fold it'll fold the active site residues into their correct locations as well. So this is interesting if you want to like potentially find a more global, or you want to, I don't know, I think it's interesting in that you could potentially land on a more active protein or a more available protein in the cell, one that could be expressed easier. But I'm sure there are other applications. In this, it's also just kind of, on one hand, just neat that you can even just port over this active site into a different protein. But one more immediately practical application, I would say, is the design of protein binders. I'm not on that slide. Why is it not over? There we go. Yes. Mm-hmm. I don't think they made this one in particular. I don't think they made this protein, but I actually am not positive on that. Right. Yeah. Right, like you would have to really put the substrate in there and then make sure you can like leave out a pocket that would fit that. There's a chance they may have done that because I mentioned in a few slides from now that they can essentially hold out a specific pocket and generate around it, but I'll talk about that in a minute from now. Yeah. So protein binders are important because we can essentially block the activity of various enzymes or receptors or viral proteins and potentially stop infections that way. And so how this would work is we can take a protein of interest that we want to essentially block the activity of. We can point out to the model the specific interface hotspots that we want a protein to generate on, and then we can generate using their model a protein that should bind to this location that we specify. So here they tested 5 different designs. They tested hemagglutinin for influenza. They tested an insulin receptor and a few other ones as well. And they got experimental data for this and showed that the binders do work fairly well, some extremely well. In their preprint, they said that they couldn't even measure the KD of some of their binders because they were just too high. But I think they resolved that in their actual publication later on. But for the influenza hemagglutinin binder, this is, I believe that's how influenza infects. I think it's the thing that actually does the infecting. I should really have known that. Essentially, they designed a binder that can then bind to a specific important part of this protein. And they got the crystal structure of this, and then they showed that it did in fact bind to the correct location in this protein, and then did fold similarly to how AlphaFold predicted it would, which is, it seems quite impressive. But one thing to note is that most of the designs that I've shown have been fairly small proteins, and this seems to be on purpose, I would say, because one of their main steps in this RFdiffusion pipeline is to get their sequences, their possible sequences that might fold into the correct structure and do the correct thing. But then they pass these all through AlphaFold, and then they see if AlphaFold also correctly matches their predicted design structure. And I believe they only tested the ones that AlphaFold also confirmed as a sort of like in silico check before going to the experimental modes. And they tested how quickly AlphaFold diverges from their predictions based on the length of the proteins that they're generating. And they found that it scales pretty exponential. It rises pretty exponentially fast based on the number of residues. And so it seems like 300 or below is more or less like the range where they agree the most. So it's possible that the proteins would have less success at the higher range, but it's kind of tough because these are comparing 2 different models, and there's no clear answer as to whether RFdiffusion is correct in these higher ones or AlphaFold is correct. But given the success of AlphaFold, it seems like we should probably put our money on that one being more correct. And so this RFdiffusion plus the AlphaFold filtering is really how they're attributing this combination of these 2 steps to the success of their protein designs. And so they previously tested those 5 binders as well in a previous paper, and they had pretty poor success rate. But here, this is their success rate using their method on number of sequences that actually worked and did the correct task at hand. And so it's, what is that? That's like 18% on the hemagglutinin out of all the sequences that they tested, which is pretty high, I would say, for considering the challenge of the problem. But they basically in the paper mentioned that because they didn't just fully attribute the success to RFdiffusion, they also said that AlphaFold, that step, the filtering step, was responsible for about an order of magnitude of the previous success rate. And they actually back calculated the success rate of their previous results if they had done this AlphaFold filtering, and saw that it did increase that success rate by an order of magnitude, or it would have. So one thing I've sort of touched on a little bit is this concept of this ligand pocket that they can specify. And so in this model, they need to, like, sometimes we need to generate around specific ligands and make proteins that can allow for ligand pockets or active sites and whatnot. And so if they don't include any specific ligand pocket, they can just try to generate a protein that would have some catalytic residues, but then it might generate into the pocket and then clash with where the ligand would go, and that's shown on the left side. And to avoid that, they can generate like a force field potential that then blocks off a certain region and then allows the ligand to hopefully occupy that space because no residues will be generated in that zone. And so this is a bit useful, but it is also a bit problematic because the ligands are not explicitly modeled, and so there's no actual chemistry being fed into the design criteria for this. It's only looking at the spatial constraints. And so this problem was solved in their next paper, like 3 months later, a little bit. So I'm going to diverge slightly, but then bring it back. But essentially, RF diffusion is Rosetta fold diffusion, and it's a generative model trained on Rosetta fold. They released, the Baker Lab released a new model a few weeks ago called Rosetta fold all atom. In this, instead of just only working with protein sequences and going to structure, they can now take protein sequence, nucleic acid sequence, metal ion, small molecules, and covalent residues as input, and then model all of these together at once. And so this, we can see an example of this here. We have a protein that's sort of in the background a little bit, but it's this is predicting the structure of the protein in complex with these nucleic acids, with a small molecule, with a metal ion, all more or less in the correct locations based on the ground truth. And then in the bottom right, we have some predicted glycosylation atoms right here. And that seems to match up as well from the ground truth fairly well. And so the reason why I'm bringing this up, actually, let me back up really quick. So like small molecule and protein structure prediction together that they're accomplishing really is also encapsulating the field of small molecule protein docking, which has been more or less explored as a separate field. And so that is really the problem of, if I have a small molecule, where on the protein will it go? And that can help us identify potential active drugs if we want to inhibit various enzymes or proteins. And so this model that can do the structure prediction altogether actually achieves this docking problem just intrinsically into the structure prediction. And they compare themselves against all the previous ML methods and find that they outperform the previous leader, which was diffdoc, a previous small molecule machine learning method. But I believe that this did not actually outperform the physics-based methods, so they just only succeed in this ML-based method. And so yeah, I bring this whole model up because you can train this further into another generative model that now takes ligands into context. And so for the same paper, they took their Rosetta fold all atom and then converted that into a diffusion model. And so here they're outlining an example of going from a small molecule to generate around that small molecule, and then create a binder that incorporates the actual local chemistry of that small molecule into the design and has potentially a better understanding of the ligand to then generate a better binder. And so they did some experimental tests on a few examples. One of them is this small molecule, I think it's pronounced digoxygenin. I might be close on that, but they took the input and then they designed a protein around it, and they measured the dissociation constant and saw that it was able to bind at a range of temperatures. And they also designed one for heme as well. And for this example, they instead of taking just the base heme, they also provided it with a coordinating cysteine residue in the initial condition that then got integrated into the protein in the final design. And they saw that it does, the design does have the absorption spectrum that they expected for if it was coordinating heme. And they found that when they knocked out that cysteine residue, then it lost that absorption, lending evidence to it actually being coordinated correctly. And then what's crazy about this design is it's actually stable at binding heme up to 90 degrees Celsius, which is like ridiculously high for a protein. And so this was kind of an achievement, but yesterday, DeepMind released an update for AlphaFold, or a results paper basically, saying that they're doing the exact same thing on this all atom structure prediction. And they released their scores on the same data set, the test data set that I showed here. And instead of getting 40%, which David Baker got, they're claiming they got 74%. So it seems like they once again just like blew everyone out of the water, which is kind of exciting and pretty neat. And I'm excited to see where this goes, because you could actually do the same diffusion model training to this new AlphaFold potentially, and then maybe get a much better protein design model than currently exists. Yes. I believe so. Yeah. So it's interesting that there's already these competitor models, and none of these are currently released yet. I think they're maybe gearing them up right now. I know the Baker Lab is planning on releasing it, but yes. Yeah. Mhm. So those are physics-based methods. So Baker was claiming that they beat out all the ML methods, and they had a different figure where they say that they're still not outperforming the physics-based methods. But DeepMind is now claiming that they're outperforming even the physics-based methods at docking, which is pretty insane. Because I think Autodoc Vina in this one that has the 52.3%, that was the previous gold standard for this task and for like drug discovery through docking, at least in the open source community. Um, so yeah, it's an exciting time for this field. I think there's a lot of growth in a bunch of different aspects, and various fields are being unified into single models, which is pretty cool to see happen live. Um, Conditional generation options are growing, and so that means that we can design for more specific cases. We can design specific motifs that we want, ligands, active sites, and so on. But there are definitely some challenges that remain in this field. The first one, which I think is quite important, is, as Klaus mentioned, we need really broad experimental validation. We need to not just consider like the fun, cool looking test cases. We need to see if these work in real life experiments, and we need to collect this data and then use it to maybe refine our models and allow it to cover more and more prediction scope and make it just most useful for actual experimentalists, which is why we're doing this in the first place. One thing as well is it'd be very important to have these models able to design around conformational changes, because a lot of proteins and enzymes require various pretty drastic conformational changes to achieve their desired function. And if we treat these proteins as just like static blocks, then we're just going to only be able to design these static blocks and not actual like flexible proteins. And the third one is more for kind of a long-term aspect, which is if we want to do therapeutic designs and have these proteins be consumed by people and go inside their body, then we need to make sure that these designed proteins are have as little immunogenicity as possible, because our bodies are quite good at detecting foreign proteins and substances and ones that could be problematic. And here we're talking about not just designing foreign proteins, but designing proteins that are even foreign to evolution as a whole. And so it's sort of a bit scary to think about like how the body would react to these like novel proteins. Like they might have catastrophic effects potentially. So one potential solution is to like fine tune these design models on just only human proteins to bias the model toward making human-like proteins, which seems like a possible avenue for moving this forward. But yeah, now we'll take some questions. But before I get to that, we created a survey on this course, this 8-lecture series. We hope you all enjoyed it. Please give us feedback on future iterations of this. I think Klaus Wilkie is planning on potentially taking this course over and integrating it into a more full-length class. And so any feedback would be greatly appreciated to make sure that the full-length class is as useful as possible for the researchers here. But yeah, thank you. Yes.